Embryonic Stem Cell Research, Genetic Engineering, and Human Cloning by Carson, Hale, Garret, Layne, and Julian Enriquez.
Genetic engineering is used by scientists to enhance or modify the characteristics of an individual organism. There are 5 steps in the process of genetic engineering. The 5 steps in this order are DNA extraction, gene cloning, gene design, transformation, and Genetic engineering is used by scientists to enhance or modify the characteristics of an individual organism. There are 5 steps in the process of genetic engineering. The 5 steps in this order are DNA extraction, gene cloning, gene design, transformation, and backcross breeding. Genetic engineering has been done indirectly for thousands of years, mostly in plants and animals, but just recently has begun to be done on humans. Dolly the sheep is a prime example of a failed attempt at trying to clone something in natural order taking place. Dolly the sheep had severe diseases such as arthritis and a lung disease. Nature doesn't kindly take to people trying to manipulate it. Human cloning is the creation of a genetically identical copy of a human. Researchers have cloned a wide range of biological materials, including genes, cells, tissues, and even animals such as sheep. The procedure consists of inserting a gene from one organism, often referred to as foreign DNA, into the genetic material of a carrier called a vector. Examples of vectors include bacteria, yeast, cells, viruses, or plasmids, which are small DNA circles.
Catholic views on human cloning, stem cell research, and genetic engineering. The Catholic Church is only against some forms of embryonic stem cell research that entail the destruction of human embryos. Stem cells, quote, have the remarkable potential to develop into many different cell types in the body during early life and growth, end quote. The Catholic Church opposes research involving human embryonic stem cells, which require the destruction of the human embryo during their isolation. In contrast, the Church has championed adult stem cell research, which does not involve human embryonic stem cells. Additionally, the recent breakthrough to reprogram somatic cells into embryonic-like induced pluripotent stem cells have further diminished the need to utilize human embryonic stem cells for research and medical therapies. The Church is not against ethically acceptable stem cell research. For instance, South Korean bishops raised $10 million for it. It does oppose, however, destroying human lives. We must respect life at all times, especially when our goal is to save lives. Moral views. The morally good. Helps with the prevention of diseases. Helps cure the incurable diseases. Provides new treatments for others. You are able to have your child how you want it to be. The morally bad. Prevents life from happening. Brings the question of what is the cost of life. Humans are taking the position of God. View on new life as a game or activity.
The ethical dilemma. Embryonic stem cell research. Embryonic stem cells offer hope for new therapies, but their use is debated and regulated differently. To obtain embryonic stem cells, the embryo has to be destroyed, which means destroying a potential human life. On the other hand, the use of these stem cells could lead to the discovery of new medical treatments that could potentially cure the suffering of many people. The debate is between the moral principle of a potential human life or curing the suffering of grown humans. Human cloning. Human cloning does not treat any diseases, but turns human reproduction into human manufacturing. The cloning procedure is so dehumanizing that some scientists want to treat the resulting human beings as subhuman, creating them so that they can destroy them for their cells and tissues. The dilemma with this is that in the cloning process, the basic relationships of the human person are ignored. An example is a woman can be the twin sister of her mother, lack a biological father, and be the daughter of her grandfather, which is against the way God wanted.
A common sense approach. It can be argued that because stem cell research fights cancer of those who are currently living, it might be considered a good thing. However, it is essentially ending a life to further somebody else's life, which is playing with God and his natural order. Scientists in higher power positions, such as a president, can build a god complex in which they think that they can do whatever they please without humbling themselves. Without a doubt, it can be seen that there are benefits to embryo cell research. However, it's clear that the consequences outweigh the benefits, and therefore it is not morally worth it to continue the progress of stem cell research. Embryonic stem cells are pluripotent stem cells derived from the inner cell mass of a blastocyst, an early-stage pre-implantation embryo. Human embryos reach the blastocyst stage 4 to 5 days post-fertilization, at which time they consist of 50 to 150 cells. A human embryo in the blastocyst stage is destroyed when stem cell research is made. Obama made a decision to further the research of stem cells around 2007. Briefly, stem cell research, only good side is that it can build a tumor to help fight cancer and save the life of current living people. However, it's obviously bad because it kills an embryo, essentially ending the potential of life.